Awesome. Cool. Yeah, we probably go ahead and get started. Hey, folks, happy Monday. How's life? How's everyone doing? Hope folks are doing well. Awesome. So in terms of course logistics, today is Monday, which means you had your second lab due yesterday at 10 p.m. So awesome. You're officially 2/9 of the way done with the labs. So woo, home stretch. In terms of course logistics, a couple of things to remind folks. So I'm gonna go ahead and open up Canvas. So just kind of keep an eye on the due dates that are on Canvas. So I have all of them for the whole quarter listed right over here. You can subscribe to this by clicking on the View Course Calendar button. And then, so Lab 2 is done. There we go. And then one other thing to make sure is just like throughout the quarter, keep an eye on your grade book. So if you go to Canvas and then go to the grade section, that's kind of like my official grade book that I'll be using to calculate grades at the end of the quarter. So in theory, all of your Prairie Learn scores should be migrating, and I think I double checked and everyone's was. But if at any point in the quarter you do something on Prairie Learn, but it's missing on Canvas for whatever reason, totally fine. Just shoot me an email with your name, PID, Prairie Learn email, all that good stuff. And I can manually update it. Other than that, I've like slightly restructured the Canvas homepage. So I had mentioned last time that I had this like list of all the bioinformatics tools that we're covering, as well as some other bioinformatics tools that you might find helpful either right now or sometime in the future when you do research or something. And then I also had this link to my online Python textbook. But then I wanted to also add this link over here to the sandbox.bio bioinformatics tutorials. I mentioned these at the end of, I think, like the first or second lecture. But I've had some folks asking, like, where can they get a little bit more practice with bioinformatics stuff and like Unix stuff. So I just wanted to put that link over here. And then it was getting a little long, so I decided to split it up into course links for things that are like mandatory for the course, and then just helpful resources that you can ignore if you want, or you can take a look at if it'll be helpful. But yeah, so sandbox.bio, I'm friends with the guy that runs it. He's got a lot of cool tutorials. Actually, our tutors are writing a tutorial that's going to be relevant for, I think, week 7's topic, or maybe week 8 or something. So yeah, they got a lot of good tutorials on terminal stuff, bioinformatics stuff. I wrote one of these. I think this is mine. Yeah, there we go. So yeah, check it out. Any questions about course logistics before we jump into today's topic? Questions, concerns, comments, dreams, aspirations? No? No? Okay, cool. So let's kind of quickly recap what we've done so far. And I'm going to go to Prairie Learn real quick, just because I think that's organized kind of nicely. So yeah, so what have we done so far? Let me go to student. Oh, geez, student view. So week 1, we just introduced, here's a bunch of interesting bioinformatics problems. Here's like how sequencing technologies work. Very high level summary of just the relevant background wet lab stuff that you're going to have to know for this class. Like how do the sequencing data get generated? And then we did a little bit of practice with Unix. And we kind of mentioned, get some practice with Unix, get some practice with Python, just kind of come up to speed with some of that background stuff. And then last week, we focused on our first actual core bioinformatics problem. So we mentioned, imagine there's this unknown pathogen, we sequence it. How do we reconstruct the entire genome sequence from those raw reads, which are each around like 100 to 150 letters long? So we talked about genome assembly, specifically a type of genome assembly called de novo assembly. I'm not going to test you on that terminology, but de novo genome assembly just means like from scratch. And then we also mentioned, okay, if I assemble this genome sequence, it's just a bunch of ACs, Gs, and Ts. That's not very meaningful to me. So then we talked about genome annotation, where we can leverage what we know about molecular biology, kind of like the central dogma of like transcription to translation, how we can leverage that information, plus stuff that we know about from existing sequences that we've collected in the past to annotate this genome sequence. So basically, to be able to figure out like from what position to what position is a gene, what gene is it, what do I think the function of that gene might be, kind of make some sense of that genome sequence. So I'll pause here. Any questions about anything we've done prior to today's topic? Feel free, and folks on Zoom, feel free to type in the chat or raise your hand. Questions, anything you want me to explain? All good? Seems like folks are good. Okay, cool. Oh, let's see in the chat. Oh, I don't think I'm sharing the slides. Oh yeah, I'm not sharing right now. Right now I'm showing Prairie Learn, but the slides are right behind it. Awesome. Okay, so now I think I'm sharing the slides, so hopefully that works. Cool. So today we're going to talk about our next kind of big bioinformatics challenge. Oh, whoops, there's a lot of people in the waiting room. There we go. Okay. Okay, so week 1, we finished by building this genome sequence. And that was kind of a hard process because we don't know what exactly we're looking for. We're just kind of blindly sequencing the sample and trying to reconstruct the genome sequence from scratch. But now let's fast forward a few weeks into the epidemic, or if we're studying some other like another pathogen that we already know about, if we're studying some species that we already know about, what if we already have at our disposal a relatively high confidence genome sequence that we can use as like a reference against which we can compare any new sequences that we collect? So today we're going to talk about another core bioinformatics problem, which is called read mapping. Specifically, read mapping, here's just a general outline of today's talk. I'm going to first introduce the concept of a reference genome. This is going to be like a high confidence genome sequence that we're going to be using as our reference against which we'll compare new sequences. We'll talk about the read mapping problem. So how do we actually go about mapping reads to the reference? We'll talk about some common types of mutations. So a little bit of review from molecular biology or genetics or something. Then we'll introduce one of the core bioinformatics problems known as the sequence alignment problem. We'll specifically be talking about like pairwise sequence alignment. And then we'll end by talking about basically the technique we're going to discuss here is going to be way too slow for us to use in practice. So we're going to talk about how modern read mappers kind of accelerate this process. And in the lab, you're going to be basically running a bunch of different read mappers. We're not going to really talk too much about their pros and cons, but you can ask the TA and the tutors about it, or you can ask me later if you want. But basically, just give you some experience running all these different read mappers that are kind of like the best practice that people usually use. So that's the outline for today / maybe Wednesday, depending on how far I get. Any questions, or should I just jump in? Folks are on board? Awesome. Okay, so let's jump right in. So first, what is a reference genome? I kind of alluded to this, but basically a reference genome is a high confidence assembled genome sequence that is constructed as a representative example of an individual organism. So everyone in this room, I'm assuming, is probably human. If you're not, that's okay. I think I said last time that I wasn't human accidentally. But basically, everyone here is human. So your genomes are almost identical. Do people have like a ballpark estimate of like roughly how identical they are? Any thoughts, like a percentage? Oh, there you go. Yeah, 99.9%. Exactly. So if I pick any two people, your genomes are going to be like roughly 99.9% identical. That 0.1% difference, roughly, is what gives rise to how everyone looks very, very different across the classroom. So if I have a high confidence genome sequence for a human, well, any other human that I sequence is going to look a little bit different from that sequence, but it's going to be almost identical. It's going to be like a useful standard against which I can compare. Same goes for other organisms. So here's just like a brief history of various different species and when their genome sequences were obtained. So the human genome sequence was actually produced in 2001. So I think that predates everybody here, which is pretty crazy to me. Wow, I was like in elementary school. Okay, so Human Genome Project ended in 2001. Technically, there's been multiple revisions to the human genome sequence. It hasn't been like completely sequenced until maybe kind of a couple years ago. They have this like telomere to telomere project. That's a whole other thing to look into if you're interested. But basically, 2001, we had a decent draft of the human reference genome. Then in 2002, mouse was the next one that we were able to obtain. Why do folks think mouse might have been a good second genome to assemble, other than the fact that they're cute? Any thoughts? Yeah, go for it. Go for it. Yeah, they're used as, lab mice, exactly. They're used in a lot of research experiments. So as a proxy to doing experiments on humans, we probably want to first do them in mouse models. And if we're doing experiments in mouse models, having a reference genome so that we could do these types of analyses would be helpful. Rat, similar reason. Chimpanzee, also similar reason. They're very closely related to humans. Dog, macaque, opossum, horse, cow. And like you'll notice, so Human Genome Project finishing in 2001, originally it started in like the 90s, and they predicted, we think we're going to assemble the first human genome sequence in like 2020. And like clearly they beat it by like 19 years. So they overestimated how long it would take. There's interesting reasons why it was able to be sped up. Actually, Vineet Bafna at UCSD was part of that private industry team that like helped assemble it. But basically, both because the sequencing technologies are becoming cheaper and higher quality, as well as the algorithms and computational tools are becoming faster, it's more and more reasonable for individual labs to be able to generate reference genomes pretty routinely nowadays. It's still like a decent effort, but it's not like this huge US federal government effort that then takes like billions of taxpayer dollars. So it's like maybe one lab can spend a few months doing it or something. So how does this tie into epidemics, and specifically the COVID-19 pandemic, which is going to be like our background for most of the lectures of this class? This paper was published in February 3rd, 2020. So this is a published paper. So it, they did the analysis, they submitted it, it went through peer review, it got published February 2020, which is when they actually published the reference genome for SARS-CoV-2. It's been revised a couple of times since then, but like barely. So they had a very high quality reference genome sequence. February 3rd, 2020, that was publicly available. For context, November 2019 was like roughly when people had any idea that something was going on. So going from like initial detection to an assembled high quality reference genome sequence in like 2 months is something that's completely unprecedented in human history. So this kind of changed the game for this epidemic, where very early on, researchers had a high confidence reference genome that they were able to use to kind of do all sorts of downstream analyses in their own local regions' epidemics. Fun fact, this guy, Eddie Holmes, is actually one of my co-authors. We collaborate a lot. He actually, in 2016, went to the wet market that we think COVID might have emerged from, and like had taken a photo of a stall that had a raccoon dog that he was like, oh shit, like something might happen here. Like this is not good how they're keeping all these animals together. So if you Google like raccoon dog Eddie Holmes, like you'll see that photo, and it's like insane that he kind of predicted it. But yeah, so that's interesting. Okay, so reference genome. So we now have a COVID-19 reference genome sequence. How do we use this to help us? So let's imagine that some person comes into our testing facility, they do a nasal swab, and we want to sequence whatever SARS-CoV-2 virus is in their nasal swab. We do the sequencing experiment. It's going to produce a bunch of short reads, and we want to then compare them against the SARS-CoV-2 reference genome that we now have. So in de novo genome assembly, what we talked about last week, we basically inferred relationships between the reads by looking at their overlap. So in other words, we were effectively comparing the reads against each other. So maybe like the first and the second read compared against each other, first and the third, first and fourth, etc. We were looking at overlaps between the reads. However, what if instead of comparing the reads against each other, we compare them against our reference genome? And again, we know for a fact that each of these reads is going to differ a little bit from this reference genome, just like we know everyone here looks a little bit different, but it's going to be almost identical. So can I compare maybe that read against this substring of the reference genome, compare this read against this substring, etc. Okay, so that's going to be like the overarching goal of this week's topic. Just a brief summary that's going to be most relevant to you throughout this week. For the lab assignment, I don't think I have you run all of these. I don't think I have you run Unimap, but basically all of these tools are different read mappers. These are different tools that, given sequencing data and given a reference genome, map out where those reads hit that reference genome. For me personally, I prefer using Minimap2, which is one of the ones that you're going to be running on the lab. It's, in my experience, the best trade-off between speed and accuracy. But there's a bunch of different read mappers that each take different approaches to obtain different accuracies and speeds. But yeah. Cool. Okay, so I'll pause here. I think I'm going kind of fast, so maybe I'll slow down a bit. I just get excited about this stuff. So any questions so far? So reference genomes, and we've set up our overarching goal of mapping reads against that reference genome. Kind of make sense? Okay, so let's talk about how we can do this. And before we do that, I want to review some common types of mutations. So common types of mutations, does anyone remember any of like the kind of core mutations from like a genetics class or mobile? What types of mutations can happen? Any thoughts? Yeah, go for it, go for it. Oh, so a single nucleotide polymorphism is basically when there's like a specific position of the genome that different people have different versions of. How does it come about? What type of mutation gives rise to single nucleotide polymorphisms? You're absolutely right, but the type of mutation has a name, and that gives rise. Yeah, go for it. Ooh, missense is a type of mutation. So missense is a subclass of what I'm going for. So missense mutation is we have a mutation that changes one amino acid in the downstream protein coding sequence, or like in the protein that we've obtained. Yeah. Ooh, a point mutation, absolutely. Yeah. Point mutation, aka substitution, is when we change one nucleotide into another. A missense mutation is one such substitution where maybe that one nucleotide change, if it's in a codon, might change one amino acid into another amino acid. You could have a nonsense mutation, which could change a codon from one amino acid to a stop codon. You could have a silent mutation, which doesn't change what amino acid it codes for. It just changes at the DNA level, but then the downstream amino acid is the same. But in short, substitution is one of the fundamental types of mutations. One letter is replaced with a different letter. Okay, what other types of mutations do we have? Ones that are less common. Yeah, go for it. Oh, perfect. Insertion and deletion. Nice. Okay, so an insertion is a type of mutation where we add one or more nucleotides, and a deletion is a mutation where we delete one or more nucleotides. There's actually way more other types of variations. There's like structural variation, so you can have like part of one chromosome breaks off and hits another one. But for today's topic, let's just assume these are the only types of mutations we care about. So substitutions, insertions, and deletions. One thing I want to mention is when these types of mutations happen in gametes, or in the case of viruses, when it happens before replication, these are heritable mutations. So when it happens, anything that comes downstream of that, whatever like offspring come about, will inherit those mutations. Okay. So questions so far? Everyone still with me? Awesome. So hopefully that should be somewhat review from like a genetics class. Okay, so with this in mind, let's imagine I have two DNA sequences and I want to align them. I want to determine where do I think insertions, deletions, or substitutions might have happened in comparing these two sequences. So maybe these are my SARS-CoV-2 reference genome versus one of my reads. Maybe this is like your hemoglobin sequence versus your hemoglobin sequence. So just two sequences that are somewhat similar. And I want to see where do I think substitutions, insertions, and deletions might have happened. This is a bioinformatics problem known as pairwise sequence alignment. And to formally define this computational problem, the input is two sequences X and Y. So for example, maybe X equals ACT, Y equals ACGT. And the output is a highest scoring alignment between X and Y. So you'll notice in the top line, I've added this gap character, this dash over here. And that is what an alignment is. I'm allowed to basically copy all the letters from both sequences, but I'm allowed to stick in any additional gaps that I want anywhere in the sequences, such that the length of these two sequences with the gaps is equal. So I could have put some in the second sequence as well. That would be totally fine. But the overall length has to be the same, and I can't put a gap against a gap. Okay, so that's kind of the setup. Okay, so is this a formally defined computational problem? If I told you here's two sequences, come up with a sequence alignment, do you have enough information to do it? Yeah, go for it. Exactly. So there's a missing piece. So the answer is, well, I got to decide on how do I score these. So I said the output is a highest scoring pairwise alignment, but I didn't mention how I can score them. So let's talk about how we can score a sequence alignment. So let's imagine that I've defined a match score, a mismatch penalty, and a gap penalty. So let's say every time I encounter a match, that's +1 points. Anytime I encounter a mismatch, that's -1 points. And anytime I encounter a gap, that's -1 points. I can define the score of a single column as one of these three values. Just is it two different letters, which would be a mismatch, two of the same letter, which would be a match, or one letter with a gap, which would be a gap. And then the score of the overall alignment would be the sum of the scores of the individual columns. So for example, let's imagine that I have these two original sequences, and then I put a gap in between the A and the C of the first sequence. I don't put any gaps in the second sequence. Let's try to score this. So what would be the score of the first column? 1, right? Exactly, perfect. +1 because it's the same letter. And what about the next column? -1, absolutely, because it's a gap. And the next column, -1, perfect. I think people are getting it. So then the last column is +1 for a match. So +1, -1, -1, +1, that gets us an overall score of 0, right? Versus what if I have this other alignment where I put a gap between the C and the T in the first sequence and I don't have any gaps in the second one? I would have +1 for a match, +1 for a match, -1 for a gap, +1 for a match. So +1, +1, -1, +1, that has a score of 2. So this is good. We've come up with a scoring metric that now visually, not even thinking technically, visually, the second alignment looks better than this first alignment, and our scoring method does indeed result in a higher score for the better looking one. Because in general, we as computer scientists have to think of how can I define some type of scoring metrics that the computer comes up with the exact same comparison that I as the human am able to just kind of like visually see. So we've done it. Scoring metric seems reasonable. Did you have a question? Yeah.
Sorry, I was wondering if you have like two alignments that are like the same score, but they're different, like X and Y. What does that mean?
Oh, good question. The question was, what if I have two alignments that have the exact same score, but they're different alignments? And I'll give you an example. Let me see if I can do this well. So let's imagine I have like X equals ACT and I have like Y equals ACCT. So let's say this, I could do that. Oops. Or I could do that. So these are two different alignments that both have a score of +1, +1, -1, +1. So both have a score of 2. So which one is better? Any thoughts? Yeah, go for it. Perfect. So based on the information we have currently, they look the same, right? They're the exact same score, and that's the only thing we've defined. And so I want to emphasize the use of the word a highest scoring alignment instead of the highest scoring alignment. There could be multiple equally highest scoring alignments, and any of them should be fine. If you're upset at me for not picking one versus the other, that means you have the problem of not telling me what is the good scoring method. According to the scoring method, these two should be indistinguishable. So any methods that I come up with to come up with an alignment should be able to return either of these and be totally okay. Yeah, awesome. Yeah, great question. Yeah. Any other questions? Feel free. Okay, so we've discussed how we can score alignments. And basically, the general idea is given two sequences, I want to find a pairwise alignment that maximizes this score. How do I do this? There's an algorithm called the Needleman-Wunsch algorithm. It's an algorithm that, given two sequences, will find a highest scoring pairwise alignment. I'm not going to require you to really understand it too in depth in this class. When you take CSE 181, you're going to implement it from scratch. So that's going to be fun. So for today, I'm going to give you a very high level overview of how it works. And the relevance for us is going to be, how does it scale? So if you don't fully take away the nitty gritty details of what it's doing step by step, I'm still going to work through it, but don't worry about it too much. Just kind of get the key takeaway of like, what is the scalability of this algorithm? That's kind of what I want you to focus on. Okay, so yeah, the details are too complex, but here's the general gist of it. So if anyone has taken CSE 101, there's an algorithmic solving technique called dynamic programming. If you haven't taken CSE 101, just jot down the words dynamic programming, and then when you take CSE 101, be like, oh, I've heard of that. But basically, this is a dynamic programming algorithm. What that means is we define the solution recursively from top down, but then we implement it bottom up, kind of from the base case upward. So it's effectively a way of, you define a recursive solution, but you implement it iteratively. Similar to like if you've ever done Fibonacci, you usually wouldn't do something like let's say if n equals 0 or n equals 1, return n, else return fib n minus 1 plus fib n minus, minus 2. You probably, if you're ever implementing the Fibonacci sequence, are not going to implement it this way because you're going to end up getting like this really, really slow, basically, when you call Fibonacci on n, you're going to call it on n minus 1 and n minus 2. When the n minus 1 is called, it's going to call it on n minus 2 and n minus 3. When n minus 2 is called, it's going to call it on n minus 3, n minus 4, and you're going to do a lot of like repeat calculations. So the idea is, let's define the solution recursively, but when we implement it, let's do something like def fib n, let's say nums equals 0, 1, while when nums is less than or equal to n, nums.append, nums -1, plus nums -2. Let me make this wider. Return nums n. So when you implement it, you're probably going to do something like this. I'm not going to ever ask you to do this because I think Fibonacci is more of like a just kind of like programming practice. But basically, instead of me like calling it recursively, what I would do is basically create a list, start by filling in the base cases, and then fill in all the subsequent values by just doing a constant time lookup of the previous values. So that's the general idea behind dynamic programming. Again, don't worry about it too much. But yeah, so for Needleman-Wunsch algorithm, what we do is we create a length of one string plus one by length of the other string plus one matrix, where the first row and the first column are like a null character. And then we put the entirety of the sequence for like sequence one as the rows, sequence two as the columns. Again, I'm never going to ask you to do this. So like if you're worried about like doing it on an exam, don't worry. I'm not going to ask you to fill out this matrix. Okay, so we do that. And basically the Needleman-Wunsch algorithm starts by filling in the top left corner as 0 as like a hard-coded value. It fills in the top row by basically saying I have to go horizontally, and horizontal is going to have the gap penalty compared to where I'm coming from. So this cell is 0 plus my gap penalty, -1 with my gap penalty, -2 with my gap penalty, -3 with my gap penalty. So I basically incur my gap penalty horizontally. And then vertically, I also incur my gap penalty to fill up the first column. So I would say gap penalty versus 0. So 0 with the gap penalty, -1 with the gap penalty, -2 with the gap penalty. And then each subsequent matrix in this cell, what I would do is basically determine which score is higher. Diagonal, where I will take the diagonal plus the match score if the two letters in this cell are the same, or I'll take the mismatch penalty if they're different, versus come vertically and take the gap penalty, versus come horizontally and take the gap penalty. So basically gap, gap, or match versus mismatch, depending on if these two letters are the same or not. So I'm going to kind of quickly fill in this matrix. Again, feel free to try to keep up with it, but don't worry too much if it kind of zooms by. There's also good videos from Pablo Pevsner if you want to kind of like check it out on your own time. But basically for this next cell, what are my options? I could either go vertically, -1 with the gap penalty would be -2. I could go horizontally, so -1 with the gap penalty would be -2. Or I could go diagonally, so 0 with the match, like the match score. 0 plus 1 would be 1. So either 1, -2, or -2. The best of these three options is taking the 1 diagonally. And I kind of keep making that decision. What are my three options? I could either go horizontally, so 1 minus 1 is 0. I could go diagonally, -1 with a mismatch, so -2. Or I can go vertically, -2 minus 1 is -3. 0 is the best option. And I kind of keep filling in this matrix using that exact same logic. I'll pause here. Questions? Concerns? Kind of makes sense. Okay. And then how do I actually get my overarching alignment? What I do is I start from the bottom right corner and I backtrack to reconstruct the alignment. How it does this is it basically sees anytime I go diagonal, let me include both of those. So let me just actually, I probably should have made slides for this part too, but I'll just do it manually. What it'll do is it'll say, okay, I'm going diagonal here. So I'm going to use this T and use that T. Now I'm over here. I'm going horizontally. So I'm going to use this G, but I'm not going to use any letters in this sequence. So I'm going to, I think I had X, the shorter one I think I had as the top one. So I'm just going to keep track of them. But yeah, so from the longer sequence, I'm using the G. And I'm not using any letters in the shorter sequence. So I'm going to put a gap. Now I'm over here, diagonal, so I'm using the C and the C. Now I'm here. Oh, geez, I can't see. Now I'm over here. I'm going diagonally, so I'm using the A and the A. And then I get to the top left corner, and that means that I'm done. Okay. So how I did that, again, don't worry. I'm never going to test you on that, but it'll come up in CSE 181. But the key takeaway here, let me erase my annotations. So the key takeaway here is the scalability of this algorithm is the length of one sequence times the length of the other sequence. Okay. Folks with me? Awesome. Okay, so that's going to be the important bit. So the scalability is linear with respect to the length of one times linear with respect to the other. Okay, so let's think about read mapping. So read mapping, I told you we have our long reference genome sequence. So this really, really long reference genome sequence. For SARS-CoV-2, this is like 29,000 letters long. And our single read is like 100 letters long. So basically, we would have to create a matrix. Oh, does scalability mean runtime? Yeah, basically time complexity. So slightly different from runtime, but same general idea. The time complexity scales as the product of the two lengths. But yeah, so if I have a short read of length 100 and I have my long reference genome of like 29,000, or if I'm dealing with humans, maybe this is like 4 billion, I would have to create a matrix that's length of 100 by 29,000 or by 4 billion, depending on if I'm doing human or not. So the problem with this approach is that, actually, I think I have slides on it. Let me hear this. So the problem with this approach is that it gets really, really, really slow, really, really, really fast. So yeah, if I want to do a pairwise alignment between a single read and the entire genome, that's going to be super, super, super slow. I have to fill in a matrix that's length of the genome by length of the read, and the length of the genome is really, really long. However, remember that we started this whole discussion by saying every single read that I collect from my sample is going to be almost identical to my reference genome, right? There's going to be some insertions, some deletions, some substitutions, but it's going to be almost identical. Okay, so let's see if folks kind of followed my matrix thing. So what would most of the things are going to be matches going to look like in that matrix? So my true solution path, even though I had to fill in the whole matrix, what does most of the things that I'm actually going to need to do are matches going to look like in that backtracking thing? Yeah, go for it. Absolutely. Do you want to see? Yeah, so exactly. So basically you saw I built this matrix. I had to fill in the whole matrix, but matches and mismatches were diagonals. So it's going to be, for the most part, along the diagonal. It might go up and down a little bit, depending on if I have insertions or deletions, but most of it is going to be along the diagonal. So any of the time that I spend kind of filling in these edges, so like this edge over here would be a bunch of insertions in one sequence, or like a bunch of gaps in one sequence, followed by a bunch of gaps in another sequence. That's going to be a really, really low scoring alignment. And same thing with this edge. All the time I waste filling in this part of the matrix kind of sucks. I'm not actually going to be using those edges when I do my backtracking, because those are going to be like really, really bad. Probably. Like most likely, I'm not going to be using those. So in other words, there might be just a few indels, but the optimal path is going to be very, very close to the diagonal. So instead of filling in the entire matrix, the thought process is, what if I only fill in things that are relatively close to the diagonal? So there's a technique that I'm going to give you a high-level description of, and you're not going to implement here, but when you get to Pavel's class, you're going to learn about it a little bit more, which is what's called seed and extend. Bless you. So doing this alignment that's okay with insertions and deletions is relatively slow. But it turns out that if I use clever data structures, if anyone took CSE 100, we learned about like Bros-Wheeler transform and some other clever data structures. It turns out if I use very clever data structures, I can find exact matches super, 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 super fast. Actually linear with respect to the read length. I can find an exact match in my genome. So the idea of seed and extend is let me take my read and let's just think about like how many insertions or deletions I think might be feasible. So a read is like 100 letters long. Let's say if I get really unlucky, maybe there's going to be like 3 insertions or deletions in my read. Well, if I split it into 4 parts, one of those parts is guaranteed to have a perfect match to the reference genome, right? And let me just kind of mention that again. So let's imagine I have a read that's like 100 letters long. So a read that's like 100 letters long. And let's say that I think maybe there's going to be like 3 insertions, deletions, maybe even substitutions, maybe 3. Maybe if I want to be extra generous, I'll say like 4 or 5. Let's say 5. Well, if I think there's going to be 5 insertions, deletions, or substitutions, I can break this up, or let's say 4, just to kind of keep the numbers clean. So if I think that there's going to be 4 insertions or deletions, I could theoretically break this up into 5 chunks. And just based on pigeonhole principle, let's say one of my insertions, deletions, or substitutions is in this chunk. One of them ends up in this chunk. One of them ends up in this chunk. One of them ends up in this chunk. One of the chunks is guaranteed to have a perfect match. At least one of them. I could end up doubling up where maybe like 2 of them fall into the same window. So maybe both of these are going to have perfect matches. But at the very least, at least one is going to have a perfect match. Okay, so what I could do is I can find exact matches of substrings of my reads very, 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 very quickly. This is called seeding. And then I can extend those perfect matches by doing just a pairwise alignment of like the immediate part close to where those perfect matches hit. So instead of doing a pairwise alignment with the entire reference genome, if I see that this read mapped, like it has a perfect match, like maybe over here, I could just do a pairwise alignment with like this window of the reference genome, which is much, much, much faster. Okay, does the idea kind of make sense? Any questions? Okay. And all those different read mappers that you're going to be using on lab 3, they're basically just doing different heuristics of how they're doing the seed and extend. So what data structure do you use to do this perfect matching? How do you go about doing this alignment that's close to that perfect match? How do you chain together multiple perfect matches? All of these things, the different tools that you're going to be using, just take different approaches to this. And that's where their pros and cons of speed versus accuracy are going to come into play. Okay. So yeah, so you use exact matching, maybe using hashes. So for example, Minimap2, it takes substrings of your reads and does like a hashing index on your reference genome and looks up those perfect string matches using that hash table index that it creates. Maybe you create a data structure like the Burroughs-Wheeler transform, which BWA uses, Bowtie2 uses. What those data structures mean, don't worry about. You can read about them if you want, but it's not going to be tested in this class. There's also some interesting stuff of like you could use wavefront or A star. I don't know if like folks have probably heard about Dijkstra's algorithm from CSE 21. So there's like the A star algorithm, which is similar to Dijkstra's algorithm, but instead of doing just distance from the start, you do distance from the start plus estimated total distance from the end. And that's how you define highest priority. If that doesn't make perfect sense, that's totally fine. Just a high level summary. This is kind of like the key takeaway of this last section. Here's some visualizations where each of these boxes is the dynamic programming matrix. And these are different approaches of how you can fill in that diagonal like best path without having to create the entire matrix. And what these are doing, don't worry, they're just kind of trippy looking and it looks like really pretty and cool. But the key takeaway is like the pattern by which they start from the top left corner and figure out how to expand. They're each taking slightly different heuristics. So this one is using Dijkstra's algorithm. This one is using the wavefront algorithm, which you don't have to worry about, but it's another algorithm that exists. This one's doing a dynamic divide and conquer. So you'll notice it's like doing substring, like half, 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 half. I wanted to get to the bigger ones again. So it does like a big portion, then like half, half, half. So it does like this divide and conquer approach. This one uses that A star algorithm. So kind of like Dijkstra's, but you're also taking into account distance from the end that you're estimating. And this one, I don't really understand how it's doing it, but it just looks pretty. I don't know. So this guy over here has like some really cool visualizations and like a nice cool overview of like what some of these different techniques are. So I have a link to it in the slides. Feel free to like read his write-up. Also, the tool that he uses to make these visualizations is open source, so you can like play around with it if you want. But yeah, I think that's my last slide. So I guess I'll pause again. Any questions about anything I rambled about? Yeah, go for it. Ooh, good question. So the question was, all of these different optimizations, are they still guaranteed to give an optimal alignment? So if like whatever that best path exists, or a best path, are they guaranteed to find it? So these specific ones are. With the read mappers that you're going to be using, the place where they start making their mistakes is that seed and extend algorithm that they use. The seed part, trying to look for perfect matches, could fail because they're gonna have like some inaccuracies where like maybe there are no substrings that they tried out that have perfect matches, or maybe the index just wasn't sufficient to find them. So usually in my experience, this is more so an optimization for that extend piece where like I have two perfect matches and I want to do a local alignment matrix just between them. And I can now also speed up avoiding filling out that entire matrix just in that section. But that would kind of look like, you could think of these as just being like the dynamic, bless you, dynamic programming matrix in between two perfect matches. But what it would look like if I had the whole sequence would basically be like diagonal lines at the perfect matches and then something like this going on in between them. Yeah, great question. Super trippy stuff. Awesome. Any other questions, concerns? No? We have like 9 minutes left. So that was my last slide. I can either end class early if people are tired and want to stretch their legs. I could show you, my plan for this week was going to be like, how can you compile these tools for your own computer? Because you're probably going to be using them at some point in your bioinformatics careers. Do you want me to start that today? Oh yeah, go for it. You have a question. Yeah, great questions. The question was, can I explain how I went from the dynamic programming grid to, okay, here's all these cool search algorithms that we can use. So basically, when I introduced this matrix, I very naively told you, start with the top left corner, fill in these, and then fill in each of these row by row. This is where it would come into play. So instead of filling in all the to figure out, instead of filling in like, so for example, over here, technically, I don't have to fill in this cell next. So to fill in a given cell, I need to know what's above it, diagonal and left. I could fill in this cell instead, right? Or I could fill in this cell. And there's actually another approach. So I think Wavefront kind of does this, or maybe I'm thinking of a slightly different one. But basically, let me actually get my whiteboard. Kind of hard to draw. Let me plug this in real quick. So another approach that you could do, and this is like one approach. So the way that I proposed filling it out, you can't really parallelize. So if you have like a multi-threaded computer, you can't really benefit from that. Oh, where's my, oh, it's on the screen. Let me send my pen to the other screen. Sorry. Come on. Tablet properties, mapping, monitor 2. Okay. Cool. Okay, perfect. So another approach is I could theoretically basically kind of go out in like diagonals like this. So basically when I'm filling out this matrix, and let me like draw a couple lines so that it's a little bit more obvious what I'm doing. Okay. It's kind of hard to draw on a separate screen. There we go. So even just with our naive approach, without using any of these clever search algorithms, I could say, okay, well, the first cell I have to fill out is this corner, which has a score of 0. Then I could fill out both of these in parallel. So I could do 2x operations and, simultaneously. And then I could fill out these 3 all in parallel. And then I could fill out these 4 all in parallel, these 5 all in parallel. So basically, if I'm going in diagonals away from the top left corner, kind of perpendicular to the direction that I'm going, each of those layers of my diagonal can technically be done simultaneously, aka if I have multi-threading on my computer, I could calculate those all at the same exact time. Does that kind of make sense? So if I have like a 64 core computer, instead of doing this 1 by 1 by 1, I can get 64x speed up roughly when I'm like in the middle of my matrix. Yeah, great question. But then those other tools don't do this. They do a slightly more clever approach of like, well, my parallelism is still calculating these edges. Like, why do I care about these? So those other approaches like figure out a clever way of maybe instead of doing all of these, let me like just do like these ones and maybe like these ones, and then maybe like these ones. Oh, but to do this one, I have to figure out these two. So it like does it a little bit more cleverly. Yeah, great question. Any other questions? No? Okay, so I think let's go ahead and stop there just because we only have 4 minutes left. So I don't think I can do anything new meaningfully. So on Wednesday, my plan was I was going to work through like showing you how I can compile these tools from the command line, just in case you want to set them up on your own computers. If anyone wants me to do anything other than that on Wednesday, just let me know. If you want me to prepare, just post on Piazza so I can like prepare something in advance. But for now, that's my goal for Wednesday's lectures. I don't have any slides. I'll just kind of work from the command line, answer questions folks have. Awesome. Yeah, thanks so much, folks. So yeah, the lab section is in 15 minutes, so make sure to go if you want to get help from the TA and tutors. Any questions from folks on Zoom before I end the Zoom call? Feel free to type in the chat. Oh, could I do more Bash on Wednesday too? Absolutely. Let me know. You can just do that day of, but just let me know like what types of stuff you want me to do. And I'm happy to like do some more Bash like practice as well. Someone asked, regarding the midterm exam, what materials or lab weeks will be tested on it? Great question. So folks should have hopefully already signed up for their exams. And let me just go ahead and pull up Prairie Learn. So the midterm is going to be about weeks 1 through 4. So it's going to cover labs 1 through 4. And I guarantee to you that every single question, whoops. Oh, I haven't added them yet. Shoot. Well, I'll do it after class, but basically I've already copied all the non-coding problems from labs 1 and 2. I'm going to do it for lab 3, and then next week I'm going to do it for lab 4 as well once we release lab 4. Every question from the midterm and every question from the final will be guaranteed to be pulled as a subset of these. So I'm not going to have any new questions. Every single problem is going to be a subset of the problems you've seen on the labs. I talked to the Triton Testing Center. They said they'll give you a non-graphing scientific calculator, but I don't think they let you bring notes. So practice, 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 get used to these problems. You can generate unlimited versions of every single problem that you want. I can type in 0 and then try a new variant. So keep practicing these and just make sure that you're comfortable with them. But there's no surprises. Every single question on both exams will be a subset of these from labs 1 through 4 for the midterm, and then from labs 1 through 9 for the final. Awesome. Okay, can I post the lab 4 material early so that the people that have scheduled their tests at the beginning of week 4 have everything? Remind me, I need my tutors and TA to test it, but I can post at least the non-coding problems. Yeah, I'll do that. So if I don't do it, just remind me and I'll do it like maybe after class. Awesome. Yeah, no problem. Cool. Okay, let's go ahead and call it there. Thanks so much, folks. Go to lab section and I will see you all on Wednesday. Bye, everybody.